Time to go to work. Nice to see you on this beautiful January day. First, there's an announcement posted about the availability of tutoring, the Biological Sciences 94 peer tutoring, which is free. And the office hours of the tutors are listed here together with their names. So let me begin with just a quick review of the topics that we talked about on Monday in order to refresh your mind and get off to a good start. So we began by asking, why is science important and powerful in the world that we live in today? And one of the explanations is that it provides a method that allows us to see the world in much greater detail, and it reveals a lot of very interesting, non-obvious, counterintuitive facts about the physical and biological world that we're a part of. So science is hugely important, and this is a theme that we'll come back to several times during the upcoming lectures. We then went on and asked, what are the characteristics of life? And your textbook actually lists five, I guess. First, metabolism, an energy system, because you've got to have a source of energy in order to allow for growth and reproduction. Secondly, a fundamental characteristic of life is that the cellular structure of life. And we talked about cells and the role of Louis Pasteur in rejecting the idea that cells might come from spontaneous generation in the last lecture. Third, there has to be a system for replication and reproduction, because it's got to be, if all cells derive from pre-existing cells, there has to be a replication process. And there also has to be a replication process to allow for population growth. There has to be an information transfer system, which is the system of inheritance. And of course, we know that in the biological world that we occupy, DNA is the primary information transfer system. And then there has to be the ability to evolve, because environments change. The world is not the same over time. In fact, one thing we're going to talk about in a few lectures, which is really a fascinating big event, was what you might think of as the first great episode of pollution on the Earth. And that was the beginnings of an oxygenated atmosphere, which was the result of biological activity. It actually turns out that the ultimate source of oxygen in the atmosphere is photosynthesis. And there wasn't, the Earth did not have an oxygenated atmosphere until the evolution of photosynthesis. And we, our distant ancestors, had to adapt to this change in the composition of the atmosphere and learn to use oxygen as part of our energetic system. So the ability to evolve. Then we talked a bit about the role of theories and hypotheses in science, and we mentioned that in theories there are typically two elements. One is a pattern. Pattern might be, for example, that we observe that all organisms are made up of cells, and then a process that accounts for that pattern. In the cellular theory, the process is that all cells derive from pre-existing cells through a process of reproduction. We talked about the cell theory, and then we began to talk about evolution by natural selection, and we'll take up that topic in greater depth in today's lecture. We then turned to the role of experiments in biology, and we considered a couple of very simple experiments. Outside of Pasteur's experiment, we also considered some relatively simple observations that rejected the notion that the reason giraffes have long necks is because they feed from the highest parts of trees, And we looked at a kind of a clever manipulative experiment to ask how desert ants actually find their way back to their nests. In both cases, those experiments were revealing. They pointed to answers that might not have been suspected before the fact. But they were incomplete in the sense that there might be other possible explanations, and what scientists do is continue to test these ideas from different perspectives until there is an overwhelming weight of evidence in support of any particular theory. And then finally, I introduced to you the topic of the tree of life. We talked about the fact that this is an exciting field of contemporary research, that the ability to sequence DNA has provided a means of looking at the genetic relationships of all branches of life and assembling them in a way that reflects how recently they originated from one another. And one of the things that I talked a little bit about was a remarkable scientist named Carl Woese, who's mentioned in your book, and who was spent almost all of his career at the University of Illinois, just died on December 30th, a man who made very important contributions to our understanding of the tree of life, because he began back in the late 60s and early 70s to try and look for mutations in small subunit ribosomal RNA molecules, part of the protein translating machinery, therefore a molecule which is common to all life, perfect molecule to use to look at relationships in all life. And in his work, he discovered that there is a very ancient domain of life called the archaea, which had not been recognized or identified before. This is pretty modern stuff. In the last 30 years, one of the three great branches of life was discovered. It wasn't something that we knew in the 19th century. So that's the basic content of the last lecture. And now what I want to do is to try a clicker question on you. This is an opportunity to see whether your clickers work, but it's also an opportunity to get some sense of the sort of questions that we'll have on the quizzes.
Ida and Kate need to test the system.
Yeah, okay, so we're going to test the system. Get your clickers out, ready to go.
Do they have to can you let them know to reset their frequency by holding the on-off button until it blinks and then pressing AA?
Do you want to
AA?
go ahead and take the mic?
Yes.
AA, okay. So you have to reset the frequency on your clicker by holding the on-off button until it blinks and then pressing A twice. Is that right?
Yes.
Okay.
AA.
So that's the first step. Okay, here's the question. Oop. What? Oh. Well, we'll have to get the question up. So every Friday, and that's next lecture, we'll have a quiz. They'll be in a format similar to this clicker question. I want you all to get perfect scores. Nothing would make me happier than to give everybody in the class an A at the end of the year. There'll be 3 questions every Friday, so we'll have 3 clicker questions. We're going to just use 1 question in this trial run as soon as we get the technology going. Are there any questions I can answer in the meantime while we're waiting? Maybe I could just continue while you.
We have to download it and start it.
Huh?
So we have to download it and start it.
Okay.
So I couldn't get in earlier because the classes are here all day.
Oh, yeah. One other thing to say is that the lectures are being recorded. There's a podcast of them, and it's got, the one for Monday has been uploaded, and I think they'll be uploaded within about 24 or 36 hours. And I will put the link to get them, to access them on the message board for you. They're going to send me the link. But if for some reason you can't come to lecture, you can bore yourself for 50 minutes by watching it on a podcast. The one lecture you really need to come to is the one on Friday when we have the quiz, okay? Because there are no makeups for the quizzes, so you need to be here for the quiz. But if you do miss a lecture outside of Fridays, you can make it up. Is there a way I can continue while you work in the background?
Unfortunately,
Yeah, it's almost done.
It's almost done. We're almost there.
Okay.
Choose
And start session.
start session. We're there.
Yeah.
all right. Good?
Did you click it?
Yeah, I did.
Oh, okay.
It's searching for using the base frequency. So,
And it's here.
Okay.
So now when you start the question, you just press play.
okay.
So it's ready.
So I'll press play.
Okay, let's see. Okay, let's go back to start.
Do you want me to?
Yeah. Okay, so here,
Okay.
well,
When you're ready to go.
I'll be darned. Maybe that got disconnected. But here's the question. Okay, you've got 45 seconds. Take out your clicker and pick the right response. Should be easy, because I just talked about most of these things. But in any case, this will allow you to verify that your clicker is working. Okay, and you can see the number of responses and the time here. Okay, so somehow we'll
This one? Yeah.
we'll let you know. Oh, here you go. Yeah, so you've all been very alert, done a good job. The right answer is number 3. Remember, I made a big point of the fact that the tree of life is drawn under the assumption that there was a single origin of all life on Earth, and one of the best pieces of evidence for that is the fact that there's a universal system of heredity in DNA and RNA. Okay, so there weren't 3 separate origins of life, but so far as we know, one origin for extant life on Earth. Okay, so let me go on now and talk about the origin of evolutionary thought. Now, it'll just be very brief, but talk just a wee bit about the history. As I mentioned in the last lecture, there were really 2 people who proposed more or less simultaneously the idea of evolution by natural selection. Now, people had the idea of evolution before Darwin and Wallis. In fact, Darwin's grandfather, Erasmus Darwin, actually proposed a theory of evolution, and Lamarck, who was a very important early 19th century French naturalist, also proposed an evolutionary scheme. The thing that was unique about the insight of Darwin and Wallis was that they proposed a mechanism, a process that would generate biological diversity, and that is natural selection. Okay? So the natural selection acts on individuals. An individual survives and reproduces or does not survive or reproduce. But the effect is on the population. And this is a point that we'll reiterate as we go along. It's populations that evolve, not individuals. Good quiz question. And this depends on individuals having heritable differences. Remember, I made the analogy to the people in the class. We all look different. In many traits, we differ. So that represents variation. Some of that variation is actually determined by our genes. Some of it is determined by the environments in which we developed. To the extent that the variation is genetically determined and that possessing those variations gives you an advantage in a particular environment, you will leave behind, you will have a better chance to survive, will leave behind your genes to the future generations. And so natural selection basically picks a non-random sample of the population based on their adaptation to the specific environment you're living in, and the genes transmitted to the next generation are a non-random sample of the present generation. That's the theory of evolution by natural selection, very simple. Now, Wallis had this insight about Wallis was an interesting guy. He had very little education. He was actually born in Wales, did a number of things as a young man. He apprenticed as a surveyor and then became fascinated with the study of beetles and natural history, and became and this was not too uncommon in the mid-19th century, he became an explorer of biological diversity. And he traveled throughout the world. He worked for 5 or 6 years in the Amazon basin, collecting biological samples, shipping those back to England. And there was a fascination in the mid-19th century with all of the exotic forms of life which were being discovered that weren't known to Europeans, that were being discovered in the New World. And people were willing to buy these specimens. So he made a living that way. Then later he spent time in Malaysia. This just represents some pictures from a trip of my own to Malaysia, and actually specifically to the island of Borneo, to a region called Sarawak. And during a period of work in Malaysia, he, it is said during a malarial fever, had this insight that natural selection was the driving force for evolution, for the creation of the biological diversity that he'd been collecting. Darwin had a somewhat different history. Darwin came from a well-to-do family. In fact, his mother's family were the Wedgwoods, and they were famous for producing expensive china. His father was a very prominent physician, and like a lot of contemporary students, his father's ambition for his son was also to become a physician. So Darwin was sent off at a fairly young age, about 15 or 16, to the University of Edinburgh in Scotland to study medicine. And it turned out that he was not a very good student. He wasn't interested in medicine. In fact, he didn't like the sight of blood. And so after a couple of years in Edinburgh, he left, then went to Cambridge and studied biology, at that time natural history. And he worked with several of the prominent figures in natural history at the time. And after he graduated, because of his diligence and interest in the subject, he was recommended to be the naturalist on a sort of voyage of discovery that was being underwritten by the British government on a ship called the HMS Beagle. It was actually a part of the British Navy. And that voyage began in December of 1831. And they circumnavigated the globe. And the route of his travels is shown on this slide. And the upper panel just shows a painting of the Beagle, the ship he was on, visiting the Tierra del Fuego. So they began at Plymouth in England and sailed down across the Atlantic southward, and they made their first stop in Bahia, Brazil. And then stopped again in Montevideo, Uruguay, and the River Plate separates, it's a huge river, miles wide, separates Uruguay from Argentina. And then down to the Falkland Islands and further south in what is today Argentina. And one of the things that Darwin was able to do was to travel inland, often for weeks at a time during these stops, and he was tremendously influenced by the fossils of Patagonia. As some of you probably know, there was a very rich mammalian assemblage in South America that went extinct fairly recently, within the last 2,000 years. So there were these fossils of large mammoths and huge ground sloths and other mammals that no longer existed in the contemporary world. Then they went around the Cape, back up, stopped at Valparaiso in Chile, eventually ended up in the Galapagos Islands. And once again, he was tremendously influenced by the Galapagos because these nearby islands had birds and lizards that were clearly related, but were different species. And one of the things that I mentioned in the lecture on Monday, and we'll come back to again, is the idea of a population. I said populations were collections of an individual in a species in the same region at the same point in time, where you could potentially have interbreeding within a population. That's an abstract notion of population. Most cases, like here, we can't define specifically the limits of a population. But in the case of islands, that's different, because the island is bounded by its shoreline. The population is also bounded, and so you've got a clear spatial definition of populations. And one of the things that was so striking was that the populations of things like ground finches were clearly different enough between islands to constitute different species. And this had a very big effect on Darwin's thinking. Well, they traveled on all the way across the Pacific, stopped at Hobart in Tasmania, stopped again in southern Australia, stopped in Cape Town in Africa, back across, touched on the coast of Brazil, and then finally, after 5 years, landed again in England. So this was a tremendous experience for a young man. And he observed things that he would never have had the opportunity to observe under any other circumstances. And there were two key influences on his thinking from the work of others who preceded him. One was Charles Lyell. Charles Lyell was the greatest geologist of his time, and began his writings in the 1820s and 30s. Darwin was very well aware of Lyell and his work, and Lyell posited that geological change was slow and continuous, and that it was the result of forces that one can observe in everyday life, okay? So geological change is slow, implying that the Earth is old. And the accumulation of materials in geology and sedimentary rocks is the result of the slow erosion of material carried, say, by rivers down into bays and deltas, where silt overlays in successive deposits through time, and you end up with strata, which are organized on a temporal sequence. So, but the key thing about Lyell is the Earth is old. The processes which have led to geological change are those that we observe in everyday life. The other key intellectual influence on both Darwin and on Alfred Russel Wallace was the writings of Thomas Malthus. Thomas Malthus was a philosopher and thinker, also a member of the clergy, who wrote a very famous essay in the 1830s called On Population. And Malthus made the observation that populations tend to grow geometrically, that is, they grow exponentially, but that the ability to increase food and sustenance can at best grow arithmetically. And therefore, there has to be a long-term conflict between population growth and the ability to acquire the resources, food and so forth, that are needed by a population for survival. So that populations do not grow at the rate at which they are capable, but at the rate which is governed by the limiting resources available to them. This means that there's a lot of differential mortality because lots of individuals in a species are born who are destined not to survive and reproduce because of this limiting availability of resources and food, okay? Well, in that, Darwin saw, and so did Wallis, a mechanism to drive natural selection. If populations tend to grow without bound, if there aren't the space and resources to accommodate all of those potential individuals, then there is the opportunity for natural selection to sort among them. And it was these two influences that played a key role in Darwin's postulation of natural selection as being the process that drives evolutionary change. So your book says there are 4 elements. Darwin broke this down into 4 elements. Individuals that make up a population vary. Some of the differences are heritable, that is, they're the result of your genes, meaning that those differences can be passed on to your offspring. The Malthusian principle, each generation, there are many more offspring produced than can survive. And finally, those individuals that survive and reproduce more offspring are not a random sample, they are a selected sample based upon the traits that adapt individuals to their particular environment. That's it. Those 4 points describe evolution by natural selection. Now, we're going to spend the rest of the lecture just looking at evidence that supports this, and we'll look at a couple of different kinds of evidence. We're going to look at historical evidence from the fossil record. We'll look at evidence for descent from a common ancestor based on what are called genetic, developmental, and structural homologies. A homology is just a similarity, and we'll explore this idea as we go along. We'll look at examples of evolution in action. Evolution is happening today. It happens all the time because the reproductive excess is being selected as we speak. So we'll look at examples of evolution in action, and then we'll talk about some of the common misconceptions about natural selection and adaptation. And finally, we'll talk about some of the limits to natural selection. Okay? So that's the rest of today's lecture. Fossil evidence. Well, what is a fossil? Fossil is simply any remains of an organism that lived in the past. And there are lots of different kinds of remains, and this picture from your text illustrates 3 different examples of different ages. Ammonite shells. Now, one of the things about fossil record is just, the record means the collective of all fossils, okay? So that's what we can infer about the past based upon the whole collection of all fossils that we know of. One of the things about fossil record is that it's biased. And we'll talk more about that in another lecture, but it's biased in favor of structures that tend to be preserved. So ammonite shells are fairly abundant fossils because a hard calcareous shell gets preserved, it doesn't break down. But here's an interesting fossil, 210-million-year-old bird tracks laid down in mud that quickly solidified and was protected. 210-million-year-old bird tracks. And then a very relatively recent fossil, just 13,000-year-old fossil of giant sloth dung. Remember, I mentioned the giant sloths of Patagonia and South America. So different kinds of fossils, different ages, make up this record of the past. Well, most fossils are found in sedimentary rock. Sedimentary rock is the rock that's formed by the layering down of sediment over long, long periods of time, millions and millions of years. And you all see, when you drive through the mountains, you'll see a cut in the road where you can see the layers of sedimentary rock. It's a common feature. Basically two kinds of rock: sedimentary, which is this layered down material from the past, and then there's igneous rock, which is produced by things like volcanic eruptions, which is new. And early on, during early geology, and up until really the 1960s or so, most of the way in which geologic events were timed was by just counting where these things were in the sediment. And based on the collection, the community of fossils within a particular layer of the sedimentary rock, they would define epochs or eons or periods of time just by using this layering as a timing device, but it didn't give you absolute time in years. Well, after radiometric dating techniques were invented, it became possible to assign absolute times to fossils. And so the old time scales are kind of an anachronism. We now know on an absolute time scale the age of most of the fossil record. And I mentioned in the last lecture that we know to a high degree of accuracy the age of the Earth, 4.6 billion years. That's a long time. But what's really interesting is that the earliest signs of life appear not that long. Within the first billion years of the Earth's history, the earliest signs of life are evident from fossil materials. And these are from things like stromatolites. So we know that life began between 3.4 and 3.8 billion years ago. Now, one of the pieces of evidence for lineal descent and evolutionary change is extinction. There are lots of organisms that we find in the fossil record that don't occur on the Earth today. So we know, we'll see an example, the giant elk. Huge elk, so big that if it existed on the Earth, we'd know it. And it doesn't, so it has to have existed at one time and disappeared. For scale, there's a human. Another example are dinosaurs, huge organisms that if they still existed, we would have seen them. So these are extinct organisms. There's no question about extinction. And in fact, we now know that extinction is a normal part of life, and that more than 99% of all of the species which have existed in the history of life on Earth are now extinct. We also have a reasonable estimate for the average lifespan of a species. It's about 2 million years. Well, now humans, maybe 400,000 or 500,000 years old, so we're only 25% of the way there on average. Maybe we'll make it to 2 million years. But it's not that long. Extinction is a natural part of the processes of life on Earth. So we see this fossils and extinction as evidence for organic change and for continuity in life. Now, clearly the fact that extinction occurs means that there's got to be a way of replenishing species. Otherwise, everything would be gone over that 3 billion year history or so. So the complement to extinction is speciation, the origin of new species, and we'll talk a lot about that in an upcoming lecture. But one of the things that is observed in studies of the fossil record are what are called transitional series, and these are series of older to more recent organisms which clearly are connected by descent. And so they represent evidence that of a common descent through time. So an example from your textbook begins with lungfish. These are lungfish at about 385 million years ago, and the focus is just on the structure that makes up the dorsal fin. Okay? And you can see in the top thing the bones that make up the dorsal fin include the fin rays, distal elements, ulna and radius, and humerus. Now it's interesting, if you follow this now through time, over a period of less than 25 million years, to the origin of tetrapods, the first four-legged organisms that actually were beginning to walk on the land. So this is a series that goes from primarily aquatic lungfish to primarily terrestrial tetrapods. And this structure has evolved from a fin, finally to a leg. But you can see the homologous portions have just been modified through time. So the ulna and the humerus trace back to the structures in the lungfish. Okay, so there's this clear structural transition in the course of evolution from lungfish to tetrapods, to four-legged terrestrial organisms, our ancestors. Now, another piece of evidence which is contemporary, which doesn't depend on analyzing the fossil record, are so-called vestigial traits. What's a vestigial trait? Well, it's a reduced or incompletely developed structure that doesn't appear to have a function or has a very reduced function. That's a vestigial trait, and we've got vestigial traits. One is pale bone. We'll see an example. So here's our relative, the monkey, with a long tail used for grasping. And here's the human coccyx, which is all that's left. It's the vestigial trait derived from, originally, the primate tail. Another example of a vestigial trait is the ability of hair to stand erect on chimps, for example. And this serves two purposes. One is for insulation in the cold. It makes the hairs of the fur stand erect. That creates more air pockets, which provides a layer of insulation, protects you from the cold. But it's also used as part of an emotional display, and aggression and so forth. Well, we still have the reflex, we just don't have the fur. So goosebumps are the vestigial end of the erect hair on things like chimpanzees. Now, there are various kinds of homologies, and I mentioned this at the start of the lecture. There's a homology is simply the similarity that exists in some structure between species descended from a common ancestor. And they can be genetic, developmental, or structural. The best homology, the most convincing homology of them all, is DNA. And because we can sequence DNA, and we can say, well, we should be very closely related to our primate ancestors, and we are. In fact, if you look at the human genome and compare it to that of the chimpanzee, we're about 98% similar in DNA. Okay, so we're very similar to chimpanzees, a little less similar to gorillas, but still quite similar to gorillas in our DNA. So that's a strong example of a genetic homology. Your book uses a different example, which is the eyeless gene from fruit flies, which has a homologous gene in the human genome, very similar in sequence, called the aniridia gene, and it is also, in both cases, they're responsible for the development of eye structures. And here's, if you sequence the aniridia gene in humans and the eyeless gene in the fruit fly, you see that in amino acid sequence, they are 90% similar. 90%. Now, you'd never get that from a random draw or from an independent origin. They're 90% similar because they descended from a common ancestor, and this is between insects and humans. So genetic homology. They're developmental homologies. And these are seen particularly when we compare the embryos of different species. So for example, you see tails and gill pouches in embryos of all chorddates, from chickens to humans and cats, because there's a common developmental progression in the program of development which leads to these homologies, these developmental homologies. So here's a gill pouch forming in the embryo of the chick. This is a human embryo, gill pouch. And finally, a cat embryo, also a gill pouch. Now, as development proceeds, that structure is lost. But during a period of development, these early structures appear transiently and then are lost. There used to be an old saying, my father was also a biologist, actually a professor at the University of California also, and he used to make this, repeat this old saying that was phylogeny recapitulates ontogeny, meaning that development is reflected in phylogeny. So that's a developmental homology. Then there's structural homologies. Most vertebrates have a common structural plan. You've got the backbone, 4 appendages. The structural plan is common, and we see that in things like limb bones, which is a very, very good example of structural homologies from birds in the center, turtle, human, horse, seal, bat, all the same structural features in terms of the bones of the forelimb. So this is a structural homology. So there are 3 kinds of homologies that I've mentioned that provide evidence that we are descended, that we are all related by common descent. The best is a genetic homology, because that's absolutely compelling. But there's the developmental homologies, which we saw in the example of the gill slits that appear during embryonic development. And finally, there are structural homologies that we see in the context of the development of forelimb in quite different extant organisms. So these are some of the pieces of evidence that have been used to support the idea of evolution by natural selection. Okay, now, is evolution actually happening at the present time? Well, yeah, we've got lots and lots of examples. Bacterial resistance to antibiotics is a great example of contemporary evolution. Insects have evolved resistance to insecticides. Just in the years since the end of World War II, the introduction of modern insecticides repeatedly are associated with a fairly rapid appearance of genetic mutations which confer resistance to pesticides in a wide variety of insects. Same thing with plants and herbicide resistance. So species are dynamic, they're not static, and when you have a strong selection pressure like a drug or an insecticide, you get a selective response. So here's an example from your textbook. This is tuberculosis. Tuberculosis is a very important disease of humans. It's an interesting disease. It's what's called a zoonotic disease, meaning that we acquired it from other animals. The thinking is that we acquired tuberculosis from living in very close proximity with cattle that as pastoral people we maintained. The tuberculum bacterium was adapted to cattle and jumped species under this close association and became an important disease of humans. And in fact, in the 19th century and up till 1950, it was one of the most important sources of human mortality. And those of you who've read authors like Dostoevsky know that tuberculosis appears as part of the story because it was a common element of life in Europe. And then a couple of things happened. One was better sanitation. More lives have been saved by public health and better sanitation than anything else probably. But one was better sanitation. But the other is the development of antibiotics, which are effective against the tuberculum bacterium. And so antibiotics began to be used widely, and then beginning in the 1980s, we began to discover cases of, not surprisingly, antibiotic resistance to drugs used for tuberculosis in the human population. And by 1993, the TB became a global health emergency again. One specific example is an antibiotic called rifampin, which actually binds to the RNA polymerase in the bacterium, and it therefore prevents the transcription of RNA. And if it prevents the transcription of RNA, it prevents the bacteria from succeeding. They die. And it's been a very effective antibiotic in the case of tuberculosis. But in recent years, mutations have arisen in the RNA polymerase, and in fact, just a single mutation, which makes the polymerase resistant to rifampin. And in fact, it's a single base pair change. The normal RPOB, that's the polymerase subunit, the B subunit of the polymerase, has a codon TCG that encodes a serine. A single nucleotide change to TTG changes the amino acid encoded to lysine and affects the structural conformation of the ribosomal polymerase and therefore blocks the effective binding of rifampin, and the bacterium survives. It's not as efficient as the normal form, but if you apply the antibiotic, you select for the mutant form, because now in this new environment with the antibiotic present, the mutant form is best adapted. And your book talks about an interesting case history, a young man in Maryland who presented himself at the hospital with fever, all of the symptoms of a very bad cold. Turned out that after a careful diagnosis, it was found that he had tuberculosis. So he underwent a treatment of, I think, about 6 months of rifampin and other antibiotics, and at the end of that period, he appeared to be okay. They released him from the hospital. 2 months later, he came back ill again. They introduced the same treatment, it was ineffective. He died within 2 weeks from respiratory failure. Why? Because in the initial treatment, almost all of the bacteria were the normal form, but there must have been a mutant or two with that serine to lysine substitution, and it was selected up. So the drug therapy killed virtually all of the bacteria, except for the resistant form, which then slowly proliferated. The guy became sick again, presented himself at the hospital, but it was too late to do anything. Why didn't it clear up? Ordinarily your immune system would deal with that small number of remaining bacteria. Why didn't that happen in this case? The reason was the guy was immunocompromised. He had AIDS. And so because he was immunocompromised, he was particularly vulnerable to this kind of event. Okay, so that's resistance to antibiotics, and we'll take up the rest of the stuff on the next lecture on Friday.